Hello, good morning, good evening, everyone. I am Vaidya Asavari Manvikar. I would like to welcome you all to GCAR Global Webinar Research Series. GCAR, Global Council for Ayurveda Research, is an international non-profit organization based out of USA. Our mission is to encourage, educate, facilitate basic, clinical, and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence-based health science globally. You can learn more about us at www.ayurvedaresearchusa.org. Please do consider joining us as a member, which will bring you access to all our resources. No nonprofit can operate and succeed without volunteers. So consider joining us as a volunteer and we can place you in a variety of projects based on your skills and your interest. Once again, our website is www.ayurvedaresearchusa.org. Now, I would like to introduce you to our eminent speaker. Welcome back, Dr. Mitali. She will be talking about third lecture, which is on Ayurgenomics.
Okay.
So you can take it from here, Dr. Mitali. Thank you so much for coming back. We are so excited to have you.
Okay. Thank you so much. And good evening, good morning, everyone. And I gave two lectures to start off what I mean by Ayurgenomics and how do we go about developing a working hypothesis to test the principles of Ayurveda. And if we are able to test the principles, then maybe we might have a better explanation on the practices of Ayurveda, because that relies on the principles of Ayurveda. So today I'll take you through the third lecture in the series. Just can you see my slide?
Yes.
Can you see it? Is it? It's okay, right?
Yes, we can see it.
Okay.
We can see the presentation. Yeah, thank you.
Okay, so today we are going to talk about molecular Trisutra through Ayurgenomics, and I'll come in a while as to what we understand by molecular Trisutra, and here try to demonstrate it through molecular axis of hypoxia, which we've been talking about in the last lecture. Basically a gene called EGLN1. It's an oxygen sensor which allows us to predict hypoxia responsiveness differences amongst Prakriti types, and a medicine called a herb called Adatoda vasica. So how we can build a framework for evidence-based solution, harnessing the principles of Trisutra through a molecular mechanism. So this is what the lecture is going to be about. So, why this talk? So the idea is basically, like I've been mentioning about Ayurgenomics, it's just a way to convert the principles into a language which could be understood in contemporary terms, so that we make it interoperable, and as a result of which some of the foundational concepts, if it can be understood, then many of the practices can be adopted through an evidence-based. So there are challenges. For example, modern medicine and Ayurveda, where both of these systems we want to bridge, there are basically ontological challenges, because one works on the principle works on mechanisms, meaning you understand the mechanism and then you use the medicine. But in Ayurveda, it is more outcome based, and the modern medicine drugs are basically come out of the premise that you give the drug uniformly, to meaning the randomized control trials does not take care of heterogeneity within the population, whereas Ayurveda medicines are individualized. So there is a very epistemological difference between the two systems of medicine. One looks at a holistic perspective. It looks at all the organs together when treatment is happening, whereas the other is more reductionist in nature and the perspective is more from organ side. So then if you want to and it is also many times one knows which is the target which is being intervened, and from there you try to build up the whole mechanism to the system, whereas the Ayurgenomics starts from looking at holistic composite prakriti phenotypes and try to elucidate molecular differences. So how these two can be reconciled, and why we need to reconcile this, and what are the kind of interesting things we can get if we are able to reconcile these two concepts, is what we are going to deal with. So as we all know, this audience, it's not just to recapitulate if there are other people in this audience, that in Ayurveda, individuals are classified by the relative proportions of the three doshas, Vat, Pitta, and Kapha. And individuals have variability with respect to what is their starting doshas. So people can be of 7 different kinds, where 3 extreme types have more abundance of either one of these doshas. So these are Ekal Prakriti, and then there are Dvandaj Prakriti, where you have Vat, Pit, Kapha, Vat, Kapha, and then there are some few Prakriti types which have equal proportions of all the three of them, which is Sam Prakriti. Now, these are assessed through phenotyping methods, which look at composite phenotypes, anatomical, physiological, psychological traits, to be able to arrive at what kind of prakriti a person has. And the baselines of these prakriti allows one to understand what is the deviation in an individual, which is described as vikriti. So what would be vikriti for Vat individual might not be so for Pith individual, they might be homeostatic doshas in there. So the idea is, if you are able to know what is your homeostatic levels of dosha, then you can understand by phenotypes of the vikriti that in which direction the doshas are perturbed. And the goal of the medicine is to bring back a person to their homeostatic doshas baseline, and not everybody to the same baseline. And this can be achieved in various ways, by medicine, by season, by different kind of attributes which are ascribed to doshas. So so we thought that the foundational, if we want to start to build then or bridge the knowledge gap between Ayurveda and modern medicine, we first need to understand dosha at the molecular level, or basically we want to understand if we can query dosha at the molecular level. So the first 2 lectures were all about how we went about developing a working hypothesis from the foundational knowledge of Tridosha, looked at these in predominant Prakriti individual using genomics approach, how we obtained multi-omic correlates with the doshas, like the biochemical correlate and gene expression correlate or genetic correlate, and were able to also find that if there are differences with respect to Prakriti, then could these differences also be seen at the molecular level? And based on that, if we see the differences, do those differences have a genetic underpinning? And if they have a genetic underpinning, whether genetic variations in them could actually associate, meaning if you have variability in those genes, would they also give differences with respect to susceptibility phenotype as is described for dosha? Then it will be a molecular picture of dosha. And if that were so, then if you know the genes, then in a model system, you can perturb the genes and you can see whether you can actually recapitulate again the phenotypic attributes of dosha. So now, since dosha is a common organizing principle, Ayurveda talks about this concept of Trisutra, where Hetu, Ling, and Aushad are connected via dosha. So you would be able to know what phenotypes you have are because of what kind of causes. And if you are able to connect the phenotypes and causes through doshas, then you can have medicines which are again understood in the context of what dosha they modulate, which then should connect to the causes and features. And this is how this whole three threads of cause, feature, and medicine are connected through a common organizing principle of dosha. So, supposing we have a molecular correlate of dosha, then would we also find such kind of a linkage coming up through that gene? So that's the acid test. That supposing we say that something is a molecular correlate of tridosha, then it should also show differences with respect to phenotype. Those phenotypes could be perturbed because of certain causes, and if you are able to link those causes and phenotypes, then you should be able to take a medicine which in Ayurveda is understood that it works in this kind of phenotype because of these causes. And so, if that was so, then does it intersect at the same axis? So then only it will become internally validated. So today talk outline how we start with one genetic correlate uh, through a genomics approach, how we validate that genetic correlate in a model system, which is asthma, and how then we study Adatoda Vasika, whether that rescues it, and whether then we can actually, if we understand the mechanistic basis, can we repurpose it in other conditions, and then if we are able to repurpose it, how does that help? And we demonstrate by this extension of this study to COVID-19, and by thereby validate the whole Trisutra. So this actually happened in a very transdisciplinary manner. The genomics started with me and Bhavna, then Dr. Anurag Agarwal. He's a modern medicine person, pulmonologist, and he also was a former director of IGIB. He had a asthma lab, meaning a lab which works on respiratory pulmonary medicine, the mechanisms and molecules and all those things. With him, we collaborated. Primarily, Bhavna was key person who was driving these studies in the Ayurveda, from the Ayurveda perspective, along with a PhD student, Atish. He is the one who primarily his work, and along with other team members, is what we are going to see. And then that was taken up as a joint project with All India Institute of Ayurveda with Dr. Tanuja Nesri. So there were other team members, but these are the primary people. You can see whether the entire spectrum of discipline was needed to be able to address the whole thing. So not I was not involved very deeply in any most of the downstream work, but this becomes a good, compelling story, and that's why I'm going to present this work with and the papers which have been referred to are also given over here. So first, if we want to start with finding the molecular correlate of dosha, then it should satisfy certain conditions. Like doshas are the dosha of an individual is governed by genetic variation, impacted by what happens in the prenatal environment. And once it goes on into an individual, it governs a phenotype of an individual, which also is dynamically calibrated based on the dosha of the circadian or the various temporal transitions, that is, different times of the day, different seasons, and different age of a person. So these doshas have a dynamicity and it is self-regulated. But if it is perturbed in a system, it leads to Vikriti, and this also becomes a signature for identifying appropriate medicine. And if you want to see whether the medicine is working, then you should see that their doshas are going back to a person's baseline thresholds. So, one such gene which we discovered and I discussed at length last time, which we found could be a candidate for a molecular correlate of dosha, because this is a oxygen sensor. It is dynamic in nature. All through the day, it governs temporal differences in hypoxia responsiveness. It also governs differences in prakriti types, and we could also show that differences in them give susceptibility to a certain condition called high altitude pulmonary edema, where Pith Prakriti people seem to have a expression and a genetic variation which predicts that they would do well at low oxygen condition, whereas Kaph individuals have a genetic makeup which kind of suggested that they might be more prone to low oxygen condition. They might have problems, and that is what we saw in a cohort of people who went from sea level to high altitude and developed high altitude pulmonary edema. So this study suggested that hypoxia axis is one of the important axes which resonates with the principles of uh, uh, doshas. And more importantly, this gene, which we discovered to be linked to high altitude adaptation, what was not only discovered in our Indian population, but it has been replicated in large number of populations in the world. High altitude populations, Tajikistan, Andes, Africa, China, Tibet, all the high altitude, this gene has been important. And in Peru, Quechua, the variability in this gene has also been linked to aerobic capacity differences. So it's a very, very important candidate, not only from what we got from Ayurveda perspective, that it differentiates Pitta and Kapha, but also it's very important as a major axis which responds to oxygen differences and can govern a large number of phenotypes. So, having oxygen and having no oxygen uh, will allow a body to switch to two different modes. Because in low oxygen condition, for example, if you go to high altitude, you need to enhance erythropoiesis, you need to enhance angiogenesis, you need to move to anaerobic energy metabolism, reduce iron metabolism, reduce cell proliferation. So all those processes, some processes need to be shut down, some processes need to be increased, and this has to be dynamically calibrated depending on what is the oxygen status, and that is what is governed by EGLN1. But what is very interesting is inherently this differs between Pitta and Kapha, as a result of which the outcome might differ if you can work in low oxygen condition, or you cannot work in low oxygen. And this also senses not only oxygen but also many other molecules. So this senses oxygen, regulates HIF, responds to different cellular cues, shows temporal variation, and also is an actionable point for many diseases where hypoxia is a cause or consequence. Variability in these axis is differentiates between prakriti, and this has systemic effects and modulates many intermediate pathophenotype in nearly all diseases, where erythropoiesis, angiogenesis, metabolism, inflammation, immunity, these are the pathophenotypes which, if you look at diabetes, you look at asthma, in any diseases it is present. So this is a very core axis which we found to differ between Pitta and Kapha. So the idea is that if this is a core axis, then is it actionable point for dosha specific formulation? So The other thing is that this gene is also not just a random gene. It is also, once this program is set, then some downstream network also gets modulated by having this variability. So another gene which actually is related to this pro-hypoxic state in pith is a gene which regulates von Willebrand factor level. Von Willebrand factor is a platelet glycoprotein which, as angiogenesis proceeds, this is made so that if there is a cut or a bruise, then it can actually attract the coagulation cascade and can plug it so that a person doesn't have bleeding. But if you have a variability in them, then it actually results in a disease called von Willebrand disease, which is a bleeding disorder. And what we observed was that with Prakriti people who are more pro-hypoxic in state has a von Willebrand allele or a genetic variation which makes them more prone to bleeding. And we asked, why is this important, that a bleeding phenotype gets associated with a pro-hypoxic phenotype? Because the genes are not adjacent to each other, but it's a functional relation. So we did some mouse experiments. I explained that in the last presentation. And we could show that if you have hypoxia, one of the important consequences, you have to have angiogenesis. In high altitude population, the hypoxia axis, which is associated with Pith Prakriti, becomes a Desh feature. And in that Desh, EGLN1, meaning the HIF1, has to be always on because they have to survive at low oxygen condition. But if you chronically keep the hypoxia axis on, then you will keep on having angiogenesis, as a result of which you will keep on making more von Willebrand factor, which will give you risk to atherosclerosis or stroke. So with Prakriti people at sea level who are more pro-hypoxic, so their HIF will be on at a lower threshold, or high altitude native have a protective phenotype in having a bleeding so that they don't die of thrombosis. So this is what we had shown in the in our earlier study, that EGLN1 is epistatic, so that if hypoxia is on, then bleeding allele of von Willebrand comes with it, and that's why Pith Prakriti in their phenotype they are more bleeding prone. So this also again kind of validates that this hypoxia hemostasis axis is something which actually differentiates Pith from Kapha Prakriti. So now from here, we also, this was what we had shown, that in a mice, if you kind of switch off PhD2, that is make it low, which is what is there in pith, then you activate von Willebrand factor level, and also the bleeding time is increased. So as a result of which, if hypoxia is on, then you have more chances of having a clot formation, not bleeding time, but clotting time. So clotting time is reduced. So now, once we have found this correlation in the human population, now can we see whether this kind of a correlation is important in disease condition? This linkage between what we find in pith-cuff axis of hypoxia, meaning EGLN1 along with hypoxia, is it relevant? So this was tested in asthma, because asthma has airway inflammation, remodeling, and local hypoxia. So this became a natural thing to test, that what happens if mouse are asthmatic and you modulate hypoxia in them, what will it do to the mouse? So this experiment was done by Anurag's group, as I mentioned, and what they showed was that if mouse has a modulation around the EGLN1 axis through a small molecule inhibitor, then what happens is, if we pharmacologically increase hypoxia signaling through a drug which actually inhibits the activity of EGLN1 so that it becomes like a pith phenotype, then the mouse becomes very sick. So the airway hyperresponsiveness is increased. Okay, and then all this collagen, fibrosis, and all of this increases as you increase the hypoxia. So this is a control, and this is each of them are suggestive of how inflammation is increased, collagen is increased, and the mouse is very sick. So these were measured at the tissue level, and this was also measured at the biochemistry level. So this, what does this suggest is that if you are maybe Pith Prakriti or a Kaph Prakriti, most probably if you are Pith Prakriti and you have an axis which is more hypoxia responsive, and you happen to be asthmatic, then there is likely to be more inflammation during exacerbation time. So from the cause, genetic predisposition, we find a hypoxia axis which can worsen asthma physiology. So now once we have this axis, this cause is between pith and kaph. This is the meaning the phenotype is between pith and kaph. The cause is hypoxia, and now the thing is that if you have the link and the hetu known, then can you actually choose a medicine which works in this axis from Ayurveda perspective and test it out, first of all, whether it can alleviate the symptoms, and then secondly, you can test out whether it actually works through this axis, because this is the axis which connects the phenotype with the causes. And this is an actionable target in many diseases. People have shown that now, if you modulate EGLN1, it is important in cartilage repair, wound healing, brain tumor, renal anemia, cardiovascular disease, myocardial ischemia, recovery from stroke, treatment of inflammatory disease. So this is the modern people are now finding that EGLN1 is a very important therapeutic target. In some, you need to make it go high, and in some, you need to actually make it go low. Low means increasing hypoxia, high means reducing hypoxia. So this is what is now proposed. So based on this, one of the medicinal herb which was picked up was Adatoda vasica. Why? Because it's traditionally used in respiratory diseases. So we have an asthma model where pith kaph axis is recapitulated at the molecular level, and it actually indicates that there is a pith kaph perturbation because of increased collagen, increased inflammation, and all those kinds of things. And Adatoda vasica is used for treating cough, bronchitis, asthma-like condition. It is important in mucus clearance, reduces inflammation. So the question was, supposing in this model we gave Adatoda vasica, will it actually go and intersect with the EGLN1 axis? So then you can close the loop of Hetu Ling Oshad. So that is what was done in a mouse, oval woman induced mouse, which is becoming asthmatic. You start to giving this drug at a different concentration, and you find that once you have oval woman induced asthmatic mice, and you have the DHB given, which is making the hypoxia more in the mice. So this is the airway hyperresponsiveness. And then you start giving Adatoda vasica, and you see how the airway hyperresponsiveness goes down, right? And then all of this is also visible at the tissue histology level, where this is the sham and this is after Adatoda vasica, and you look at all these kind of inflammation, fibrosis, bleeding, all these kinds of things, phenotypes, which are typical phenotypes in inflammation in the lungs, Adatoda vasica is able to reverse them. So then it also, in an earlier study, it was meaning in this study it was shown that when it reverses by actually HIF and alpha. It reduces the concentration of hypoxia, uh, the transcription factor, which switches on everything in hypoxia. It actually inhibits that, having the D2, which dexamethasone is also not able to reverse. So steroid resistant asthma, in that also Adatoda vasica reduces hypoxia. It is seen at the ELISA, at the protein level. You can look at the expression of the protein going down, and then you actually look at the expression here, and then what it also shows is that it actually moves the cells. It restores mitochondrial dysfunction, which happens during asthma, and it restores mitochondrial function, and it also restores mitochondrial shape. So mitochondria becomes elongated, or it has a different shape. It actually tells whether the mitochondria are functional. So at those levels, also at the cellular level, it was shown that the way Adatoda vasica actually inhibits hypoxia. So in hypoxia, as I had mentioned, anaerobic respiration mostly happens. So it switches it from anaerobic to mitochondrial TCA cycle. So the mitochondrial shape is restored, mitochondrial function is restored, and what even if dexamethasone doesn't work, Adatoda vasica works over there. So this was one important study where it actually showed that through the Trisutra framework, Adatoda vasica can now be traced to molecular axis of EGLN1, and mechanistic validation of dosha indicated formulation acting through the same axis that defines the dosha correlate. Extract of Adatoda vasica possess anti-hypoxic properties and can reduce severe airway hyper inflammation induced by hypoxic response in a treatment-resistant asthmatic mice. The anti-HIF1 alpha effect of AV also restores cellular hypoxia mediated loss of mitochondrial morphology and function. So, having seen this loop that hypoxia is an intermediate pathophenotype, and Adatoda vasica works in modulating that through the EGLN1, then Adatoda vasica will it also work in other diseases where hypoxia is seen? So, can that work in other places also? Because if it is to follow the Hetu Ling Aushad, and ideally from Vikriti phenotype, if you think that again it's a pit cough perturbation, and supposing it causes hypoxia, would Adatoda vasica work there also? So to be able to do that, so this is if this is a true intermediate pathophenotype and Adatoda vasica works actually through this, then other places like sepsis or even in COVID-19 like condition where hypoxia was a major kind of a cause was seen, would Adatoda work there? So Atish then tried them out. So one of the model is a pulmonary fibrosis model, which is induced by bleomycin. It induces hypoxia, which raises HIF1 alpha, which raises more fibrosis, leading to more hypoxia. So this kind of a model was made, and in that Adatoda vasica was given, and you can see that this TGF beta concentration, which is actually a sign of inflammation, with bleomycin, it gets elevated when Adatoda vasica is given. The levels are reduced, and HIF1 alpha, which is indicator of hypoxia, is also reduced. And this is also seen, the fibrosis which is seen in bleomycin, actually this is restored in Adatoda. So one of the models of hypoxia of bleomycin-induced thing, this is collagen secretion staining, which is seen over here. So you can actually see that Adatoda vasica works here. Then the second model which was used was a sepsis model. So this is cecal ligation and puncture, where this is basically you cause a polymicrobial sepsis after cecum is tied and punctured, so that gut bacteria leaks into abdominal cavity and it causes a septic response. So in this model, again, cyclosporin is one of the medicine which is given, and this is the Adatoda vasica. You can see the HIF1 alpha is given over HIF1 alpha is seen, concentration is seen in this, and then this is the cyclosporin and with Adatoda. So in the naive mice, nothing happens, but you can see in the IL-6, which is again an inflammation marker, how cyclosporin it is reduced, and also Adatoda vasica is able to bring it back to that level, and IFN gamma is somehow increased in Adatoda vasica, which again shows that there might be some other mechanisms which also is very different from cyclosporin. But what was also very interesting is that they looked at mortality of after giving Adatoda vasica. So the bleeding is reduced over here with cyclosporin and with Adatoda vasica, which is increased after the cecal ligation. And this is the extent of mortality reduction. Percentage survival is much more when you have cyclosporin and when you have Adatoda vasica. So these are the number of hours after survival. So Adatoda vasica also ameliorates hallmarks of lung inflammation and injury in mice model of this. The third thing is, as I had mentioned, when you have hypoxia, then thrombosis is also one of the consequences because of von Willebrand factor increased because of angiogenesis in hypoxia. So this was also tested, and what was seen was that if you have hypoxia increased, then von Willebrand factor, which is elevated in increased hypoxia, is reduced when Adatoda vasica is given. And HIF1 alpha, it inhibits HIF1 alpha. So basically, Adatoda vasica works through modulating HIF1 alpha. It's an inhibitor of HIF1 alpha, because of which it also stops the coagulation related characteristic and also platelet activation. So this was again shown with the hemostatic outcome of hypoxia response induced by inducing EGLN1 activation, which meaning by EGLN1 inhibition by an siRNA, so that hypoxia increases, that can be ameliorated by Adatoda vasica. So it also works on the hemostatic outcome. After that, we looked at what happens when Adatoda vasica is given to the lung expression at the RNA level, which will also tell what all pathways are really getting modulated. And one of so these are typically the pathways. So all the details are given in the paper. So these are upregulated genes and these are downregulated genes when Adatoda vasica is given and when the lungs were taken out, and we tried to see what are the expression differences as a consequence of treatment with Adatoda vasica. We see that it favors expression of genes which are important in immunity, adaptive immunity, and inhibits genes which are involved in hypoxia associated angiogenesis, fibrosis, and inflammatory cascade. So whatever we see at the gross phenotype level, by airway hyperresponsiveness, then at the tissue level and at the biochemical level, we also see a similar picture at the cellular level. So all these ones are like preclinical models of COVID. So during the time these experiments were being done, it was primarily propelled by that if Adatoda vasica works in hypoxia and in COVID, all these phenotypes were seen. An impaired hypoxia responsiveness leading to coagulation, leading to inflammation. So would Adatoda vasica work over there? So we needed to test it out in preclinical models, which work through hypoxia, and if it works there, then it's like an intelligent guess that it is likely to work in COVID. Moreover, many phytochemicals in Adatoda vasica actually, when docking was done, not only binds to multiple proteins which are linked to hypoxia and inflammation response, for example, TGF beta, HIF1 alpha, HIF1 gamma, L6, JAK1, TNF alpha, JAK3. These are all the major players of inflammation and hypoxia, but it also bound to many of the target proteins of SARS-CoV-2. So these are all the phytochemicals of Adatoda vasica, and there are certain phytochemicals which are also binding with very good binding energy. So it shows that because of its polypharmacological nature, it can at the same time bind to a large number of players in the host as well as in the virus. And there was a in vitro study done to see whether it can inhibit viral replication, and it was shown that nearly 60% of the titer can be also reduced by having Adatoda vasica given to cells which are infected with SARS-CoV-2. This was an in vitro study. So, putting all these things together, this paper provides this figure and explains it in greater detail that there are many things which happen during SARS-CoV-2 at the earlier stages and at the severe stages like fibrosis, severe airway, acute lung injury, blood clotting. At the molecular level, people have seen all of them. And then at the genomic level, many of these pathways are deranged. At the cellular level, immune cells, platelet counts are compromised, and at the tissue level, airway hyper inflammation is compromised. And as I showed you, at all these intervention points, we see that Adatoda vasica in various model systems have shown that it is likely to work in COVID. So this is where this was the preclinical study which came out in respiratory research. Primarily, Atish is a pharmacologist, and other students worked, and Bhavna was one of the key persons. She is an Ayurveda doctor, Anurag, and I was there. And so this study showed that there is a possibility that this could work in SARS-CoV-2. So based on this, because COVID was ongoing, a clinical trial was planned with All India Institute of Ayurveda. It was a randomized, open-label, three-armed trial where the drug formulation was given in a 14-day intervention study, and there were three groups made. One where Adatoda vasica was given, one where Tinospora cordifolia was given, and one in which the combination was given. And the study happened for 14 days, and at all times, many parameters were studied as to the body temperature, how many days it comes down, viral clearance by RT-PCR. So this was registered as a trial, and at the level of at the biochemical level, it was at the molecular level, it was seen that if you had Adatoda vasica, it brings down hypoxia, HIF1 alpha, ferritin angiogenesis, and PT-INR is increased so that you are reducing risk for thrombosis. Tinospora shows more of an immunomodulatory effect. And when you have the group C, then there is a quicker viral clearance, and also some of the markers which are linked to severity are reduced. So this was the paper which was published, which actually showed that Adatoda vasica and Tinospora, both of them could ameliorate clinical study, but in the context of Adatoda vasica, it actually suggested that it could act as a modifier of HIF1 alpha in COVID-19 patient with reduction in the levels of ferritin, VEGF, PTINR, which are markers of hypoxia, inflammation, and thrombosis, which highlights the potential use of this in a during an ongoing infection, right? So these are all the various things which were studied, and these are, like, for example, for CRP levels, it was down by in all the treatment. Some of these D dimer levels, you can see the group A, it is lesser. So this is all the summary of all these studies. You can go back to this paper, and you can see HIF1 alpha is also reduced, VWF is also reduced when Adatoda vasica is given. It is not so when with Tinospora cordifolia. And then there is VEGF levels also, which are kind of modulated. So this now you can see that first we have genetic correlate. We go to the mechanism. Based on mechanism, one can plan out what are the intervention, and then if that works through the Trisutra network, then you can use it for other conditions which are similar, and also in a condition which could be labeled as Anukt Vyadhi in Ayurveda. And the ultimate proof is actually taking it right to clinical trial. So if one molecule could go till that level through the dosha linkage and the Trisutra format, there are so many more things which can be done through this Trisutra framework. So basically what this validates, it validates a mechanistic basis of dosha specific formulation with through the Trisutra framework. Adatoda is no longer just a traditional thing. Its efficacy is testable to a specific molecular axis, and it can be applied to other formulations in a similar manner. It also validates that Ayurveda medicines could work through these intermediate pathophenotypes which are correlated to doshas. For example, bleeding is one intermediate pathophenotype which is correlated to Pitta Prakriti. Cell proliferation is an intermediate pathophenotype which is correlated to Vata Prakriti. Similarly, so this is something which is again hypoxia causes intermediate pathophenotypes of inflammation, thrombosis, or bleeding, depending on what your genetic makeup is. And so, if you know that something works through hypoxia axis, it would be able to modulate all the other conditions in which similar phenotypes are seen, which could also be assessed by Vikriti Parikshan. The third is it provides a evidence-based solution for an Ayurveda setting where if a person is presented with such a kind of a molecular marker, and if that has connect to Ayurveda from the dosha linkage, then one can actually think of how to source proper medicine, or how to use proper preclinical model for providing evidence-based solution. And finally, it works in an Anukt Vyadhi condition. COVID-19 would not have description in Ayurveda text, but the fact that this framework is a generic in nature with a definitive molecular axis, one can actually use the Anukta framework to actually use the same medicine for an unknown condition or a condition which is not given in Ayurveda. So this is like an Anukt Today's date, most of the disease, if they are described in modern terms, how would it make Ayurveda sense? It can this such kind of a molecular study can become assistive in trying to deconvolute a modern description of disease in Ayurveda language so that one can actually source better medicines from Ayurveda for more effective outcome, and one can also identify the hallmarks through which these medicines work. So there are assay systems possibility which would tell that if you are giving this medicine, if it is working through such a kind of a axis, then these are the things which are measurable, which will actually provide an evidence based solution in Ayurveda. So this is just a template. This is a framework how one can move from one correlate to a mechanism to a preclinical, and it can actually be true for many other things. And the same three sutra axis, now one can also, if this works at the molecular level, you can start from a drug and try to find out the mechanistic basis, hetu and link, and again have this, if it is internally validated, then you can approach the problem from looking at the phenotype, looking at the causes, looking at the drug. One can actually try to discover more and more interventional points or treat more and more diseases. So I will close now, and in the next class, I will show another aspect of the using the Three Sutra framework. This time from taking a drug axis. So this would be a drug which is used in multiple conditions. How is it used? Can we actually use again the Three Sutra framework to be able to find the causes between ling and hetu, so that if there is a newer hetu or newer link, can that same medicine be repurposed. So thank you all for patient hearing, and I'm open for questions.
Anybody has any questions for Dr. Mitali? Thank you so much, Dr. Mitali, for such informative and valuable, presentation. It was amazing. So you explained like how to bridge the gap between Ayurveda, modern medicine. You explained to how to validate Ayurvedic concepts by using Three Sutra framework, genomics, clinical trials, the studies on mice having hypoxia due to asthma, and also how to use Aratoda Vasica. That was absolutely phenomenal. And ultimately, we want to use this framework for the Anukta Vyadhis because there are so many new diseases, Anukta Vyadhis, which have not been explained into Ayurvedic text. I mean, you cannot see the, correlate, but how to use all this information and the framework, that was absolutely wonderful. It will be very beneficial for, all the Vaidyas and, practitioners.
Yeah, if you have time, I'll relate a small story how it matters
Mhm.
to me. This knowledge. I recently had a problem in my house. My mother was very ill, and she was in ICU, and she had similar phenotype which we see in mice. uh, she had edema. She was on ventilator. She had inflammation all over her body. She was not able to breathe. Mucus, lots of mucus was there, and it was a very poor state she was in. And somehow I was thinking, and in an ICU critical care setting, you cannot actually think about how you would introduce an Ayurveda medicine, right? But I was
Mhm.
thinking here, there is a all the phenotype which were visible was just like what we saw in this mouse model, in sepsis model in COVID-19, because she also had an ongoing sepsis going over there. So I talked to the doctor over there, and then I kind of asked whether we can try something in Ayurveda, and he was very open. So I approached Dr. Nitin Kochar. All of you know Dr. Nitin Kochar, And I asked him whether there's something like Adathoda, because I have seen such amazing results in Adathoda. So is there a possibility? And she had lots of inflammation, so one could not see her wrinkles and everything. So he thought for a while and he gave a medicine, Vasa Arishta, which is the Arishta preparation of
Mhm.
Vasa. And
Mhm.
he said it could be given through rice tube and we can try. And they were very accommodating and they started giving her. And in 3 days, she was out of ventilator and she was moved to a private ward. All her wrinkles had come back, her inflammation had gone. But we did not know, they did not know, because we were not measuring, or we did not know whether this worked or other concomitant medicine worked, or it was a willpower. So it was at that. But something had worked. Otherwise, the way she was in and the way the modern doctors also were open to having this medicine given, it was something which for me, this kind of a, if this kind of an evidence is there, then I would actually have a rational way of thinking and convincing a person that maybe this is likely to work. And maybe I have a thinking that that is the one of the things which really worked during that condition when our body was fully in edema and all those kinds of things which were there in a sepsis. So there is so much to do, and the problem is, how do you, if we don't develop enough evidence, and if we don't develop enough number of biomarkers, and her, and everything was getting monitored because she was in ICU and she had this medicine for nearly 2 weeks till there was another infection, and it was all her medicines were changed or whatever stopped. But uh, her, nothing happened to her kidney, nothing happened to liver, all those kinds of things. But the problem was that since it is not, a trial has not been conducted in a modern clinical setting, how do you uh, have the liability framework decided, which actually make people hesitate to try out uh, Ayurveda medicine, specifically in a clinical care setting, right? So I had to
Right.
give a consent that I am responsible for this medicine being given with the full knowledge of telling my family members. So we have to change this situation, and that's where the whole community in an interdisciplinary manner have to participate together to see how we can increase the options in a integrative medicine setting if such a kind of a thing works so wonderfully. So that is an open thing. We don't know. It was one n of one study, but it gives me a hope that there is a possibility that this kind of a thing can be attempted in much more kind of a way where documentations are done to be able to analyze and see whether in such situation, because people like to take Ayurveda medicine at leisure. In critical conditions such as an ICU, nobody would like to take a risk because that time everything is very uncertain, right? But if
Right.
Arishta preparation work in emergency condition, then that's a potent thing, because that's also an alcohol extract of a Ayurveda herb along with other combination, which also makes it bioavailable, reduces overall inflammation. So there is a mechanistic understanding now attached to it. So that's it. So I just wanted to share this also this thing in this forum.
Thank you so much, Dr. Mitali, for sharing real life experience of your own. I mean, you have to really believe, trust into it so much so that you are ready to, take this risk. And also, it is also important to see the openness, by the allopathic doctors who are working in the ICU, to be able to integrate this into their practice and see. And I'm so glad that Dr. Nitin Kochar, he gave you, this idea and guidance that was really, really valuable. Thank you so much. This is amazing. So this is how it should work, right? Like in the future, Because ultimately the goal is to heal the person, to cure the person and come out of the situation. And if it is being used in the ICU setting where it is not, like only a chronic disease, because typically Ayurveda is used in the chronic diseases, but when there is this kind of situation which is, very acute and difficult, so life-threatening, where Ayurveda is being used along with allopathic medicine, that's absolutely amazing. Yeah, no, thank you again. Thank you so much. Yeah. So
Okay.
this is like absolutely phenomenal, and your teamwork has been amazing. Just, the best team you have got. Thank you,
Yeah. Thank
everybody.
you.
And yeah, thank you for sharing all your experience and explaining it to everybody. So all the attendees, thank you so much attending our webinar. And this is being recorded. So this will be on the YouTube channel. So definitely go ahead and check our YouTube channel, view her previous presentations as well as the other webinars too. And please subscribe our YouTube channel, like, comment, share the videos. Once again, I invite you to become also member of GCAR and also fill out the volunteer form. You can now also subscribe to our IU update newsletter to stay tuned with our projects and events. And if you have any questions, please feel free to conduct and get in touch with us at Ayurveda Research USA at gmail.com, or you can reach out to me at asavari at mnayurveda.com. Thank you so much.
Thank you. Thank you.
Namaste. We'll meet in the next session. The next
Yeah.
session will be the last Saturday, the same time of next month in August. So please mark it on your calendars, and I would invite everybody to join us for this movement. Dr. Mitali and her team has done such a valuable work, and we all should actually bring it, in our practices too. So yeah, please join us definitely for our next lecture. Thank you again.
Thank you.